I want you to picture a string, but not just any string. A specific, impossibly long molecular string.
We're starting with the scale again, are we?
We have to. I mean, we really have to, because I found a number in the notes for this deep dive that just, it completely derailed my understanding of biology for a little while.
Let me guess. It has a lot of zeros.
It does. 2 times 10 to the power of 11 kilometers.
It's a number so large it just sounds fake. It doesn't feel real.
Exactly. For anyone trying to visualize that, it's roughly the distance from the Earth to the sun.
Yep.
And back.
Right.
And then to the sun and back again. And again, maybe 60 or 70 times.
And just to be clear, that is the aggregate length of all the DNA inside a single human body.
Yeah.
If you could somehow unspool every single strand from every single cell and lay it end to end.
Yes. But here's the paradox. Here's the part that I just could not get past when I was reading through our source material. That's Leninger's Principles of Biochemistry and Harper's Illustrated Biochemistry. That string isn't just floating in empty space. It isn't, wrapped around some giant spool in a factory. It's inside you. And more specifically, about 2 meters of that string is inside a single cell nucleus.
A nucleus which is what? Maybe 5 to 10 micrometers across?
5 to 10 micrometers. I mean, that's microscopic. So the analogy that kept jumping into my head, it's like you have a piece of fishing line, really thin fishing line, that's long enough to stretch from New York City all the way to Los Angeles.
Okay, I'm with you.
And your job is to fit that entire cross-country string inside a single tennis ball.
And crucially, not just stuff it in there.
Right. That's the key. If I just crammed a cross-country fishing line into a tennis ball, it would be a hopeless, tangled mess. I'd never be able to find the start or the end, let alone a specific inch in the middle.
But if the cell has to do that, it needs to find a specific gene, a tiny segment of that string instantly.
Instantly. And it needs to read it, copy it, and sometimes repair it, all without the whole system knotting up into a useless ball.
And that is the packaging paradox. That is really our entire mission for this deep dive.
Welcome back to the deep dive. Today we are moving away from the world of what the code says, the A's, C's, T's, and G's we usually obsess over, and we're looking at the engineering. This is all about architecture.
I think that's such an important distinction to make right at the top, because usually when people think about genetics, they think about the sequence. They picture DNA as like a text file on a computer screen. It's just flat. Just information.
But it's not flat. It's a physical object. The thing.
It's a massive physical object that is subject to the laws of physics, of thermodynamics, of tension, and chemistry. It's a library, for sure, but you have to imagine it's a library that has to fit inside a marble.
Okay.
And inside that marble, the books are constantly being opened, closed, copied, and repaired 24/7, all without the shelves collapsing or the books getting torn.
And before we get into the mechanics of those shelves, we should lay out the ground rules. As always, we are synthesizing 2 very dense, very important textbooks. Leninger gives us that beautiful structural logic, the physics of the folding, the why.
And then Harper's Illustrated Biochemistry grounds us in the medical reality. Why does this matter? What happens in disease? It gives us the clinical pathology.
Exactly. And we need to say this every time. This is an interpretation. It's a guide. We are trying to build you a conceptual map, but we are not replacing the map itself.
You still have to read the chapters. If you're a medical student or a serious student of biology, reading the source material is non-negotiable.
Wow.
We're here to give you the framework, the scaffolding, so that when you read about histone octamers, your eyes don't glaze over.
Right. You see the engineering brilliance behind it.
You see the why.
So let's bring back our narrative frame. We've been using Medlock Holmes, our biochemical detective. In past deep dives, he was, looking for typos in the script. He was tracking down metabolic crimes.
Right. He was tracing the flow of energy, following the carbon.
But today, the detective work is totally different. He's not reading the book. He is inspecting the library itself. He's looking at the glue holding the pages, the design of the shelves, the layout of the rooms.
Because if the library is built wrong, or if the shelves collapse, it doesn't matter how perfect the books are.
You can't read them. Or even worse, the books themselves get damaged.
So where do we start? At the most fundamental level of packaging.
Let's start at the bottom, the fundamental unit. We have this 2-meter thread of DNA. It's naked, it's vulnerable. And frankly, from a chemical standpoint, isn't it kind of stiff?
That is a fantastic point that often gets overlooked. On a molecular level, the DNA double helix is actually quite a rigid molecule. It has what's called a high persistence length.
Meaning?
meaning it doesn't want to bend tightly. It wants to stay as a straight rod. If you just left it alone, it would be this long, stiff thing.
So we have a stiff rod that we need to somehow coil into a microscopic ball. How on earth do you force it to pack down?
You need a spool, a very, very strong spool. And you need a force powerful enough to overcome that natural stiffness and bend it. And this is where Leininger introduces us to the first level of compaction, the nucleosome.
The beads on a string model, I remember this from like high school biology, but I have a feeling we need to upgrade the resolution on that image.
We definitely do. If you were to gently extract chromatin, which is just the name for the whole complex of DNA and protein, and you look at it under a powerful electron microscope, it literally looks like beads on a string. The DNA is the string, and the beads are these little protein clusters.
But cluster feels too messy, too random.
It is. These are precision-engineered machines. Each bead is a complex called a histone octamer.
Octamer, octo meaning 8.
Exactly. It's built from 8 protein subunits. You have 2 copies of a histone called H2A, 2 copies of H2B, 2 of H3, and 2 of H4. They all come together to form this sort of cylindrical hockey puck shape.
Okay, so we've got a protein puck. What happens with the DNA?
The DNA wraps around the outside of this puck, and it's very specific. It wraps about 1.65 times around the core.
1 2/3 of a turn.
And that segment is about 146 base pairs of DNA long.
I want to pause on the word wrapping, because we just established that DNA is stiff, and I also know that DNA has that famous phosphate backbone.
Yes, it does.
Phosphate is negatively charged. And from basic physics, negatively charged things repel each other. So if I'm trying to wrap a negatively charged cord around a spool...
It should fly right off. It shouldn't stick.
Exactly. It should repel itself and fly off. So what's the glue? What is holding it there?
Electrostatics. This is one of those moments where the chemistry is just so simple and so beautiful. It's almost poetic.
Okay.
If you look at the amino acid sequence of those histone proteins, the H3s, the H4s, all of them, they are incredibly rich in 2 very specific amino acids, lysine and arginine.
Sure.
Positively charged, and not just a little bit. At the pH inside a cell nucleus, they carry a strong positive charge. Their side chains are basically little positive magnets.
Ah, I see it now. So the spool itself is a magnet.
A powerful one. The histone core presents this massive positive surface charge. The DNA backbone has its massive negative charge. They slam together with incredible electrostatic force. And that attraction is what overcomes the physical stiffness of the DNA and forces it to bend into that tight coil.
That makes so much more sense. It's not just passively sitting there. It's being actively held in place by a powerful magnetic grip.
A very strong grip. And it's not just any old coil. Leininger points out something very specific and very important here. The DNA forms a left-handed supercoil around the histone octamer.
Why does the direction matter? Left-handed versus right-handed? What's the difference?
Oh, it matters a great deal for the physics of accessing the information later. Think about a rope or a cable. If you twist it one way, you tighten the strands, making it harder to pull them apart. If you twist it the other way, you can actually introduce some slack. The DNA double helix itself is a right-handed coil. By wrapping it in a left-handed direction around the histone, the cell is essentially underwinding the DNA. It's storing what we call negative supercoiling tension.
Which is useful. Why?
It's like preloading a spring. Later, when you need to pull the 2 DNA strands apart to read a gene or to replicate the DNA, that stored tension actually helps the strands separate. The box pops open easier when you unlock it.
Wow.
If it were wrapped the other way in a right-handed supercoil, you'd be tightening the helix even further, making it that much harder to open up and read.
So it's an assist mechanism that's literally built into the storage system. That's incredible.
It's a form of stored energy. It's brilliant. Now, looking at this whole nucleosome bead, there's one more player we need to introduce. We mentioned the octamer, the 4 pairs of core histones, but there's a 5th type of histone.
H1, the lonely histone.
The linker histone. H1 is different. It doesn't sit inside the bead with the others. It sits on the outside. Leininger's description is perfect. It's like a clip or a seal. It binds to the DNA right where it enters the spool and right where it exits.
So if the octamer is the spool that the thread is wrapped around, H1 is like the little piece of tape you put on the end to keep the thread from unraveling?
That is a perfect analogy. Without H1, the DNA would breathe a little, like it'd slide on and off the spool. H1 clamps it down tight. It seals the whole structural unit and helps pull adjacent nucleosomes closer together. It really locks the wrapping paper in place.
You know, when we talk about these proteins, histone H4, histone H3, they can sound like, I don't know, random part numbers from a catalog, just another protein in the cellular soup. But there was a fact in the source material that absolutely floored me regarding just how important these specific proteins are. It involves a pea plant and a cow.
Ah, yes. The evolutionary conservation. This is, and I don't think this is an exaggeration, one of the most profound facts in all of biochemistry. It really puts the deep in deep dive.
Lay it out for us, because it's astounding.
So humans, cows, pea plants, we are obviously very different organisms, eukaryotes, but very different. We diverged from a common ancestor with plants over 1 billion years ago.
1 billion years.
Since then, almost every single gene we have has changed significantly. Mutations happen, they accumulate. That's how evolution works.
1 billion years is a very long time for typos to accumulate in the script.
An immense amount of time.
Yeah.
But if you take the gene for histone H4 and you look at the amino acid sequence it codes for in a pea plant, and you compare it to the sequence in a cow.
How many differences are there out of how many amino acids?
2.
Just 2.
2 amino acid differences out of a total of 102 in over 1 billion years of separate evolution.
That seems statistically impossible. It's like 2 people independently writing the same 1,000-word essay and only having 2 typos different between them.
It's not chance. It's the most intense selection pressure imaginable. Think about what that actually means. It means that the nucleosome, this specific design for wrapping DNA around a positively charged puck, is so perfect and so absolutely critical to the survival of eukaryotic life that nature has not been able to improve upon it.
Or, to put it another way, if you change it, you die.
That's exactly it. Any mutation that occurs in the histone H4 gene that changes the shape of that puck even slightly or messes up the precise placement of those positive charges, well, the DNA doesn't wrap correctly.
The chromosomes don't pack.
The library collapses. The cell can't function. It's a lethal error. So the only versions of this gene that have survived for 1 billion years are the ones that have remained almost exactly the same.
It's the fundamental operating system of all complex life. You can change the apps, you can be a cow or a pea or a human, but you absolutely cannot change the BIOS.
That is a great way to put it. You do not mess with the kernel.
Okay, so we have our beads on a string, we've taken our long 2-meter fishing line, and we've wrapped it around these little magnetic hockey pucks. That shortens the length, right?
It does. It gives you about a 7-fold compaction.
7-fold is nice, but we need to fit that cross-country line into the tennis ball. We need way, way more than 7-fold compaction.
Oh, yeah. We need to get to about 10,000-fold compaction by the time we have a visible chromosome, so we are just getting started.
What happens next? Do the beads just sort of pile up on top of each other?
They coil into a rope. This is the next level of structure, often called the 30-nanometer fiber. The beads themselves start to interact with each other.
How?
Those little H1 linker histones we talked about are key here. They help pull the nucleosomes together, causing the beads on a string to coil up into a much tighter, thicker spiral or solenoid.
So the string becomes a thread, and now the thread becomes a rope.
And then the rope gets organized into loops. This is where we get into what's called higher order structure. And this is an area where the textbook view has really shifted in the last decade or so.
What was the old view?
The old view was basically just more coiling. You coil the rope, then you super coil the coil like an old school telephone cord, and just keep packing it down until it's a solid block.
And now, what do we think now?
Now, thanks to some really clever modern imaging and molecular techniques like something called Hi-C, we know it's not a random packing. It's organized into very specific, discrete loops. The official term is TADs.
TADs? That stands for topologically associating domains.
It's a mouthful, I know. But the concept is actually pretty intuitive. Imagine a TAD as a distinct neighborhood or maybe a single room within the library.
Explain that analogy.
You have a very long strand of that 30-nanometer fiber. The cell machinery grabs 2 points on that strand, which might be thousands of base pairs apart, and it pins them together at the base. This forces the DNA in between them to bulge out into a big loop.
What does the pinning? Who are the bouncers at the door of this room?
A set of proteins. A famous one is called CTCF, and it often works with another complex called cohesin. They act as the anchors, or the staples, or the rings in a 3-ring binder that hold the base of the loop together.
Okay, so I've got my DNA organized into these loops. Why is that better than just a condensed rope?
2 main reasons: regulation and organization. Think of the loop as a private workspace. Everything inside that loop can interact with each other very easily. A gene can physically bump into its regulatory switch, its enhancer, if they're both trapped in the same confined space.
But that gene probably won't interact with the enhancer in the next loop over.
Exactly. It prevents crosstalk. It's like putting up walls between different departments in an office. You don't want the activate liver function switch to accidentally drift over and turn on the grow a neuron gene just because they happen to be near each other on the linear sequence. The TAD structure keeps them physically segregated.
So it's not just about saving space, it's about information security and creating functional neighborhoods.
It's both. And it gets even more organized. These loops are then themselves anchored to a massive internal protein skeleton inside the nucleus called the nuclear scaffold or nuclear matrix.
The scaffold. Leninger has this amazing image. If you use enzymes to chew away all the DNA and the histones, you're left with this ghost-like shape of a chromosome.
That's the scaffold. It's the internal framework of the library shelves. It's what keeps all those loops organized in 3D space so they don't become one giant tangled mess.
And this leads us to an even bigger concept, which I love because it just shatters the cartoon image I had in my head of a nucleus. The idea of chromosome territories.
It's a fascinating and relatively recent discovery. We used to think the nucleus was just a soup, a big bowl of spaghetti where all the chromosomes are mixed up together.
But that's not the case.
Not at all. It is highly territorial. Chromosome 1 has its own designated spot in the nucleus. Chromosome 2 has its spot. They don't really mix or overlap much. It's like a map of a country. California doesn't just wander over and decide to sit on top of Florida.
And Harper adds a really cool functional layer to this. The address of a gene. Its location within the territory and within the nucleus actually matters.
It matters a lot. Real estate is everything, even in the nucleus. The center of the nucleus is generally the active zone. It's rich in the transcription machinery, the enzymes that copy genes into RNA.
So busy chromosomes, the ones that are being read a lot, hang out in the prime real estate downtown.
Exactly. They will extend loops of their DNA into the center of the nucleus to be transcribed. And the quiet ones, the ones that aren't being used,
But they get pushed to the suburbs.
they get shoved to the walls. The periphery of the nucleus, right up against the nuclear envelope, is the storage closet. Inactive DNA is often anchored there, kept out of the way of the main action.
So the cell is constantly rearranging the furniture. It's like, okay, I need this file. Let's bring it to the center table. I'm done with this one for now. Let's shove it back in the corner.
It is an incredibly dynamic, living machine. The architecture is constantly being remodeled based on the cell's needs. It is absolutely not static.
But wait, I see a physics problem here. A big one. And this is where my brain starts to hurt a little bit every time I think about it.
I love physics problems. Lay it on me.
We are talking about coiling and looping and twisting and moving these incredibly long ropes of DNA. Have you ever tried to coil up a garden hose? Or an old-school telephone cord?
I know exactly where you were going with this.
If you twist one end, the other end knots up. You introduce tension, torsional stress, it wants to snap back.
That phenomenon is called supercoiling. And in the context of DNA, managing that supercoiling is a matter of life and death for the cell.
Why is it so dangerous? It's just a bit of tension.
Because if you take the DNA double helix and you try to pull the two strands apart, which you have to do to read a gene or to replicate the DNA, you are overwinding the DNA further down the line. You are tightening the coil ahead of the fork.
Okay.
If that tension gets too high, the physical strain will literally shear the DNA. It will snap the phosphate backbone.
So the simple act of reading your own DNA could shatter it.
It absolutely would, unless you have the molecular magicians.
The magicians, you mean the topoisomerases?
Topoisomerases. These are enzymes whose entire job is to manage the topology, the shape, the knots, and the stress of the DNA.
How do they work? Because when I read the mechanism, it sounds completely insane.
It is wild. Let's start with the simpler one, type I. Type I, the topoisomerase finds a tense, supercoiled piece of DNA, grabs onto it, and then it cuts one of the two strands of the double helix.
It cuts the DNA on purpose. It breaks its own genetic code.
It makes a single strand break, but it holds on tightly to both ends of the cut. This allows the other intact strand to pass through the break, which lets the helix spin around and release the tension. It's like letting a twisted phone cord spin until it's straight again.
And then it seals it back up.
And then it chemically religates or seals the break perfectly.
Okay, that's bold. But type 2 is the one that really scares me.
Type 2 is the heavy artillery. It's for when you have a bigger problem like two different DNA helices tangled around each other in a knot.
So how does it solve that?
Type 2 grabs one of the double helices, And it cuts both strands, a complete double-strand break.
It severs the chromosome clean through.
Clean through. This opens a temporary gate in the DNA. Then it physically passes the other intact double helix through that gate. And then once it's through, it glues the first chromosome back together.
That is like performing open-heart surgery with a chainsaw just to untangle a knot. The risk seems enormous.
It is. And it's an active process. It requires ATP for energy. It's a very high-risk, high-reward operation. Because if that enzyme fails for any reason, if it cuts the DNA and then drops it before it can reseal it,
You're left with a double-strand break.
which is one of the most catastrophic types of DNA damage, the chromosome literally falls apart.
And this is exactly why Harper highlights this as such an important clinical target, right?
Precisely. This mechanism is so essential, especially for rapidly dividing cells, that we can exploit it as a weapon.
A weapon against cancer.
And against bacteria. A lot of our best antibiotics, like ciprofloxacin, target the bacterial version of topoisomerase, which is called gyrase. The drug lets the enzyme make the cut, but then it freezes it, preventing it from resealing the break.
So the bacteria essentially ends up shredding its own DNA, trying to untangle it.
It commits a kind of forced suicide. It introduces so many breaks into its own genome that it dies.
And the same principle applies to chemotherapy.
Yes. Drugs like doxorubicin or etoposide do the same thing to human topoisomerases. Cancer cells are dividing like crazy, so they're constantly using their topoisomerases to untangle their DNA as they replicate it. The drug sabotages the enzyme, turning the cell's own maintenance crew into its executioners.
That is a brutal but incredibly clever therapeutic strategy. It really underscores how fragile this whole architecture is. You don't even need to attack the genes directly. You just have to break the tools that manage the folding and unfolding.
That's it. If the library's shelving collapses, the information inside is lost one way or another.
Okay, so we've built the library, we've organized the shelves into these tab loops, and we've got the maintenance crew, the topoisomerase managing the tension. But a library isn't very useful if every single book is open on the table all the time. You need a system to decide what to read and when.
This brings us to segment 3, access control, the permission system of the genome.
And we have two fancy words that are central to this, euchromatin and heterochromatin.
The Greek roots are actually helpful here. EU means true or good. Hetero means different.
So true chromatin and different chromatin, what is the actual physical difference?
It all comes down to density. Your chromatin is lightly packed. If you look at it under a microscope, it stains lightly. It looks pale and diffuse. This is where the DNA is accessible, the beads on the string are spaced out, the 30-nanometer fiber is looser, the books are open on the table.
And this is where the active genes are, the genes that are being turned on.
Generally, yes. This is the transcriptionally active part of the library. Heterochromatin, on the other hand, is the opposite. It's dark, it's dense, it's tightly compacted, the nucleosomes are smashed together, H1 is locked down. This is where genes are silenced.
So if I'm a liver cell, for instance, I don't need the genes for how to be a retina. I never need to read that book.
Exactly. So your liver cells will take those retina-specific genes and pack them away into dense heterochromatin. You basically zip file them and throw them into deep storage where they can't be accidentally opened.
But Harper makes a really important distinction here that I found very useful. Not all heterochromatin is the same. There's constitutive and facultative.
This is a key concept. Think of constitutive heterochromatin as being like structural concrete. It is always packed. It never, ever opens up. It's part of the permanent structure of the chromosome.
And what are some examples of that?
The best examples are the centromeres and the telomeres. These are structural parts of the chromosome, the handles and the end caps. You never need to read the genetic information in a handle, so the cell keeps them permanently crated up to protect them and maintain their structural integrity.
Okay, so that's the permanent storage. What about facultative?
Facultative means it's optional or situational. This is DNA that is packed away tightly right now in this particular cell type, but it could be opened up if the cell were to differentiate into something else, or vice versa. It's dynamically regulated.
And the absolute best, most intuitive example of this is the calico cat.
It is the absolute best visual aid in all of biochemistry. If you own a calico cat, you own a living, breathing diagram of facultative heterochromatin.
Walk us through this. How does the packaging of DNA create a patchwork cat?
Okay, so it all starts with the sex chromosomes. In mammals, females have 2 X chromosomes, XX. Males have 1 X and 1 Y, so XY.
Right.
The X chromosome is quite large and has thousands of important genes on it. The Y chromosome is tiny and has very few.
So if you just left it at that, females would have a double dose of all the genes on the X chromosome compared to males.
They would. And in biology, gene dosage is critical. Having double the amount of a particular protein can be just as bad as having none. It can be toxic. So mammals evolved a very clever mechanism to fix this, called dosage compensation.
The cell has to shut one of the X chromosomes up.
Exactly. Very early in the development of a female embryo, when it's just a small ball of cells, every single one of those cells makes a random choice. It picks one of its two X chromosomes, either the one from its mother or the one from its father, and it completely and permanently shuts it down.
How does it shut it down?
It condenses it. It packs it into incredibly dense, facultative heterochromatin, crumbling it into a tiny, dark-staining ball called a bar body that gets pushed off to the side of the nucleus. It is silenced for the life of that cell and all of its descendants.
And the choice of which X to shut down is completely random in each cell.
Totally random. In one embryonic cell, it might silence the mom's X. In the cell right next to it, it might silence the dad's X.
So now let's bring in the cat.
Okay. The gene for fur color in cats is located on the X chromosome. And let's say there are two versions, two alleles of this gene. One version codes for orange fur, and the other one codes for black fur.
And our female calico cat has one of each. She's heterozygous for this trait. She inherited an orange X from one parent and a black X from the other.
Precisely. So in the embryonic cells where the black X chromosome happened to stay active, all the skin cells that descend from that one will produce black fur.
And in the patches of skin that grew from the embryonic cells where the black X was inactivated and turned into a bar body,
the orange X is the only one left running the show, so that patch of fur will grow orange.
and because this random choice happened very early in development, when there were only a few cells, those cells then divided and grew into large patches of tissue.
Exactly. So when you look at a calico cat, you are seeing a living physical map of which X chromosome was packed away and silenced in that particular region of the skin. You are literally looking at facultative heterochromatin with your naked eye.
That is just, it's so cool. But it begs a huge question. How? How does the cell actually do that? How does it lock the door on a gene or an entire chromosome? We talked about the magnetic grip, the positive histones, and the negative DNA. How do you turn that magnet off and on?
Now we are getting to the heart of epigenetics. We're back to what's called the histone code.
The code that's written on the spools themselves, not just on the DNA string.
Remember we talked about those histone proteins having tails?
Yeah.
The N-terminal ends of the proteins kind of stick out from the main bead.
The little wiggly bits.
The wiggly bits. Those tails are like control panels. A whole host of enzymes can come along and attach different chemical flags or modifications to those tails. And adding or removing these flags changes the physics of the chromatin.
Okay, give me the most important one. The main on-off switch.
That would be acetylation.
So adding an acetyl group to the histone tail, what does that do?
The acetyl group gets added to the side chain of a lysine amino acid. And it does something very simple and profound. It neutralizes the positive charge on the lysine.
Wait a minute. The positive charge is the glue. It's the magnet that's holding the negatively charged DNA onto the spool.
Exactly. So if you neutralize that positive charge, you effectively turn off the magnet, the electrostatic grip loosens, the DNA relaxes, and can even push away from the spool a bit.
The library shelf opens up, the book becomes accessible.
And the transcription machinery can get in and read the gene. So as a general rule, histone acetylation equals gene on N.
And if you remove the acetyl group, desulfurylation,
An enzyme called histone deacetylase, or HDAC, comes and snips it off. The positive charge on the lysine comes back, the magnet snaps shut, the chromatin condenses again, gene O off.
it's like a dimmer switch for gene expression.
It is, and it's more complex than that. You could also add methyl groups, methylation, which is more nuanced. Sometimes it signals on and sometimes all off, depending on which amino acid gets methylated. It creates binding sites for other proteins that can then lock things down even tighter or recruit activating machinery. This whole system is the heart of epigenetics.
Above the genetics.
Yes. Because this information is heritable, but it's not in the DNA sequence itself. When that liver cell divides, it doesn't just copy its DNA, it also copies the pattern of histone modifications. It passes on the instructions to the daughter cell saying, "We are liver cells, keep the I genes locked down."
So your cellular identity isn't just your sequence, it's your bookmarks. It's how the library is annotated.
That is a beautiful way to put it. It's the layer of interpretation on top of the raw text.
We've covered the basic storage unit, the nucleosome. We've covered the higher level organization, the loops and territories. And we've covered the access control, the heterochromatin, and the histone code. Now I want to look at the specialized anatomy of the chromosome itself. the special parts, the ends and the middle, telomeres and centromeres.
The structural heavyweights. These are the parts that are less about information and more about physical integrity and mechanics.
Let's start with the centromere. In all those classic cartoon drawings of chromosomes, the little X shapes you see in textbooks, this is the pinched-in waistline where the two arms of the X meet.
Right. But it's not just a waistline. It's the functional handle. It's the anchor point. When a cell prepares to divide, it builds this incredible scaffold of protein ropes called the mitotic spindle. The job of the spindle is to grab onto the chromosomes and pull the copies apart into the two new daughter cells.
And the centromere is where those ropes attach.
Just the docking site. If the rope can't attach properly, the chromosome gets left behind.
Which means you get cells with the wrong number of chromosomes, aneuploidy, which is a hallmark of many genetic disorders and almost all cancers.
A very dangerous situation.
So how does the cell's machinery know this specific spot is the handle? Is it a specific DNA sequence that says attach here?
In humans, it's a bit more complicated than a simple sequence. It's usually made up of lots of highly repetitive satellite DNA. But the real crucial marker isn't the DNA sequence itself, but rather a special kind of histone that's only found there.
Another histone variant.
A variant called CENPA. It's a special version of histone H3. The cell only incorporates CENPA into the nucleosomes at the centromere. It acts as an epigenetic beacon that says, build the kinetochore complex here. This is the spot. Attach the ropes here. It structurally defines the center of the chromosome.
Got it. So it's a structural marker, not a sequence marker. Now, let's go to the ends, the telomeres.
The aglets of the chromosome.
The little plastic or metal tips on your shoelaces?
It's a perfect analogy. Telomeres are protective caps at the very ends of our linear chromosomes. In humans, the sequence is very simple. It's TTAGG. Repeated over and over and over again, thousands of times. This sequence doesn't code for any protein. It's a sacrificial buffer zone.
Why do we need a buffer? Why do we need to sacrifice it?
Because of something called the end replication problem. This is a fundamental flaw, a design limitation, in the way our DNA polymerase, the copy machine, works. It needs a little RNA primer, a platform, to sit on before it can start copying the DNA.
Okay, so it needs a running start.
It does. But when it gets to the very, very end of a linear piece of DNA, on one of the strands, there's no place to put that final primer.
It's like a painter who needs to stand on the floor to paint the wall. When he gets to the very last inch of the wall at the bottom, there's no floor left for him to stand on to paint that final bit.
An excellent way to put it. The consequence is that every time the DNA is copied, the new strand is just a little bit shorter than the original template.
So with every single cell division, our chromosomes get shorter?
Yes. A little chunk of the end is lost.
Yeah.
So if we didn't have telomeres, we would start eating into our actual important genes. We'd lose critical information with every division. So instead, we have this long buffer of meaningless TTG repeats that we can afford to lose.
But eventually, that buffer has to run out.
It does. And when the telomeres get too critically short, the cell's damage-sensing machinery recognizes the frayed ends as a form of DNA damage. This sounds the alarm. My ends are exposed. This is dangerous. And that signal triggers the cell to enter a state called senescence. It retires. It stops dividing permanently.
This is the biological aging clock.
It is one of the primary drivers of cellular aging. Most of our cells have a finite number of times they can divide. This is called the Hayflick limit, and it's largely dictated by the initial length of our telomeres.
There's a loophole, right? An enzyme called telomerase.
The fountain of youth enzyme. Telomerase is a remarkable machine. It's a reverse transcriptase, which means it carries its own little RNA template inside it, and it uses that template to rebuild the TTAG sequence, adding new repeats onto the ends of the chromosomes.
It refills the tank.
It refills the tank.
So if we have this amazing enzyme, why do we age? Why don't our cells just keep telomerase on all the time and divide forever?
Because immortality is incredibly dangerous. In most of your somatic cells, your skin cells, liver cells, heart cells, the gene for telomerase is turned off for F.
Why would the body do that?
It's a safety mechanism. It's an anti-cancer mechanism. If a cell can divide forever, it has more opportunities to accumulate mutations. A cell with unlimited divisions that also picks up a mutation that tells it to ignore stop growing signals is the definition of a cancer cell.
So we want our normal cells to have a forced retirement plan to prevent them from becoming cancerous.
Absolutely. The trade-off for a longer lifespan is a higher risk of cancer. So our bodies evolved to keep telomerase off in most tissues.
Which means that cancer cells
Cancer cells, in order to become successful, almost always have to find a way to mutate themselves to turn telomerase back on. In about 90% of human cancers, telomerase is reactivated. It's how they hack the system to achieve immortality.
And that's why telomerase inhibitors are such a huge area of cancer drug research.
A massive area. If we can find a way to safely turn off the tap to inhibit telomerase in cancer cells, then those cells will start to age out and die just like normal cells.
It's a fascinating biological trade-off. The process of aging protects us from cancer, but then, eventually it gets us anyway.
Biology is always about trade-offs.
Yeah.
There is no free lunch, especially not in genome architecture.
Speaking of the contents of the genome, let's talk about what's actually in this library. We have all this elaborate packaging, the histones, the loops, the protection, but what percentage of this entire library is actually readable books? What percentage of our DNA actually codes for protein?
This is a fact that shocks a lot of people. It's the 1.5% reality.
1.5%.
That's it. Only about 1.5% of the 3 billion base pairs in the human genome consists of exons, the sequences that are actually translated into proteins.
That is shockingly low. I mean, if I bought a book and only 1.5 pages out of every 100 had a story on them, I'd want a refund. What on earth is the other 98.5%?
Well, historically, we had a very simple and very arrogant name for it: junk DNA.
Which we now know is one of the biggest misnomers in the history of science.
Extremely. We're slowly figuring out what it all does. A big chunk of it, for example, is introns. These are non-coding spacers that are located inside of genes.
It's like editing a movie.
That's a great analogy. The cell transcribes the whole gene exons and introns together into a long RNA molecule. Then a process called splicing comes along and cuts out the boring bits, the introns, and splices together the action sequences, the exons, to make the final message.
And why have that extra step? Why not just have the exons back to back?
Because it allows for alternative splicing. You can edit the movie in different ways. By choosing to include or exclude certain exons, you can create many different proteins from a single gene. It massively expands our proteomic complexity.
So that's useful. What else is in the 98.5%?
Transposons. Lots and lots of transposons.
Leninger calls these parasitic DNA elements.
And that's pretty much what they are. These are sequences often derived from ancient viruses that have the ability to copy themselves and then jump or insert themselves into new locations in the genome. They make up nearly half of our entire genome.
We are half. Genomic parasite.
Genetically speaking, yes. We are hosts to a massive ecosystem of ancient viral-like elements that have been hitchhiking in our genome for millions of years. Most of them are now dead fossils, unable to jump anymore, but the remnants are still there.
But Harper makes the point that even this junk, these transposons and other repetitive sequences, might have a crucial purpose.
A structural purpose. Remember the tennis ball. Remember the need to organize the DNA into specific TAD loops. To do that, you need the right spacing. You need filler material. You need structural concrete between the important bits.
So if you deleted all the so-called junk, the architecture would fall apart.
The spacing would be wrong. A gene might end up too close to an enhancer it's not supposed to talk to, or too far away from what it needs. The loops wouldn't form correctly. The whole regulatory landscape would be thrown into chaos.
So the junk is actually the scaffolding and the spacing and the insulation that makes the whole complex architecture possible.
Exactly. It's not junk, it's infrastructure. And we're finding more and more that a lot of it is transcribed into regulatory RNAs that fine-tune gene expression. The ENCODE project was a massive undertaking that showed that the vast majority of our genome is biochemically active in some way.
We've covered a lot of ground, but I want to end by focusing on the clinical reality. We've hinted at it with topoisomerase and telomerase, but let's get specific. What happens when this elegant packaging system fails?
We see the profound fragility of the system. Let's start with DNA repair. Your DNA is under constant assault from UV light, from the sun, from chemicals in our environment, from oxidative stress, just from breathing.
How does this cell's repair crew find a tiny bit of damage in this incredibly dense packed library?
Needs a flare, a stress signal. And that signal, amazingly, is another histone variant, a special version of H2A called H2AX.
A2AX.
When a double-strand break occurs, the worst kind of damage kinases come and add a phosphate group to all the H2AX histones in the chromatin for thousands of base pairs around the break. They chemically light up.
So the damaged site becomes a glowing beacon.
A massive beacon. This phosphorylated H2AX acts as a landing pad that recruits the entire DNA repair machinery.
Yeah.
If you don't have that beacon, or if the repair machinery itself is broken, you get devastating diseases.
Like xeroderma pigmentosum. XP?
XP is a heartbreaking example. It's a defect in a system called nucleotide excision repair, which is the machinery that's supposed to fix the damage caused by UV light.
So for these patients, sunlight becomes literally toxic.
Exactly. The UV radiation causes adjacent thymine bases in their DNA to fuse together, creating something called a thymine dimer. Normal cells can just snip these out and replace them. In XP patients, that damage goes unrepaired.
And that leads to?
It leads to an incredibly high rate of skin cancer, often at very young ages. Their library maintenance staff is missing this specific tool for sun damage.
And what about the BRCA genes? We hear about BRCA mutations constantly in the context of breast and ovarian cancer.
BRCA1 and BRCA2 are key players in the team that fixes those catastrophic double-strand breaks using a process called homologous recombination. If you have a faulty BRCA gene, your cells, particularly in the breast and ovaries, struggle to repair those breaks correctly.
So the genome becomes unstable.
It accumulates mutations and large-scale chromosomal rearrangements at a much higher rate. That instability is what drives the cell towards becoming cancerous.
There was one more example in Harper that I found really subtle and fascinating, hemoglobin lipour, which is caused by unequal crossing over.
This is a fantastic example of how a disease can arise because the library is actually too organized, or at least too repetitive.
How does that work?
Well, many of our genes exist in families or clusters. The genes that code for the different parts of hemoglobin sit right next to each other on the chromosome. And importantly, they have very similar DNA sequences because they arose from gene duplication events long ago. During meiosis, the process that makes sperm and egg cells, the chromosomes from your mom and dad have to line up perfectly so they can swap pieces of DNA in a process called recombination.
Right.
But because these hemoglobin genes look so much alike, sometimes the chromosomes get confused. They misalign. It's like buttoning up your shirt, but you're off by one buttonhole.
I have definitely done that.
So when the crossover and swapping happens, you don't get a clean trade. You end up fusing the front half of one hemoglobin gene to the back half of the neighboring one. You create a new, hybrid Frankenstein gene. That's what hemoglobin lipour is. It's a fusion protein that doesn't work very well, and the person gets a type of anemia.
So it's a disease caused not by a typo in the letters of the book, but by a physical misalignment of the shelving during a renovation.
That is a perfect description of the mechanism.
So if we synthesize all of this, we started with a string long enough to reach the sun. We ended up with mismatched buttons on a shirt. What is the new worldview for our detective, Medlock Holmes?
I think the key takeaway is that the mystery of the genome is not just in the sequence, the one-dimensional string of letters. It is in the topology, the 3D structure, the folding, the architecture. DNA is not a static hard drive. It is a dynamic, breathing, shifting piece of molecular machinery.
And the so what for the listener? The take-home message.
The take-home is that we are now entering the age of epigenetic medicine. We are moving beyond just sequencing genes to figuring out how we can manipulate the folding and the access to those genes.
We're developing drugs that can change the shape of the library.
We are. We have drugs called HDAC inhibitors, for example, that block the enzymes that remove those gene-on-acetyl marks. They can force tightly packed chromatin to open up. We're testing them to see if we can force cancer cells to reopen and read the books for genes that tell them to stop dividing. We are, in a very real sense, learning how to be the librarians.
That is a very powerful image.
And I'll leave you with this final thought to mull over. Every single cell in your body, your neuron, your skin cell, your liver cell, has the exact same DNA, the exact same library of books.
Right. The same genome.
So who decides? Who is the master librarian that walks into the embryonic neuron and says, okay, for you, we are gluing all the liver-specific books shut forever, and then walks into the liver cell and does the exact opposite?
That is the billion-dollar question of developmental biology.
It is. It tells us that your identity biologically is not defined simply by the DNA you have. It is defined by how you fold it.
I am never going to look at a tennis ball or a spool of thread or a calico cat the same way again.
Mission accomplished then.
Thanks for diving deep with us. We'll see you in the next library.